dorms at our facility. We have an 18-acre complex with dorms and a bunch of different laboratory spaces as well. From an operational perspective, we have over 115,000 acres of easily flooded woodlands, wetlands, and open grasslands. So in Florida, we have about 90 different species of mosquito. In St. Johns County, 50 roughly. From a global perspective, there's over 3,000 different species of mosquito. They all have their own unique biology and behavior, so control can be challenging. Some of the mosquitoes will oviposit their eggs in dry soil that will be flooded. So salt marsh mosquitoes. So if you live on the intercoastal areas of, Duval, St. Johns County, the salt marsh mosquito emerges in the millions and they can fly up to 30 miles and their eggs can stay viable for years. Whereas what maybe you encounter in your backyard, Aedes aegypti, Aedes albopictus, are important vectors of dengue, chikungunya, Zika, yellow fever. Those eggs can also stay viable for years in your potted plant containers, your dog bowl, until it gets a rain event, irrigation event, or you just add water to it, it flushes those eggs down, and then they will develop from there. Culex, our West Nile virus vector, they lay eggs directly on the water surface. And so when we implement control, different strategies are used depending on their biology and behavior. We have 60,000 acres of protected area. Essentially, any of those areas in this map that have this red hashtag are protected lands where we have specific arthropod management plans. And this is through the Florida Department of Environmental Protection. We're regulated under the Florida Department of Agriculture and Consumer Services, but we have to work with our partners and our stakeholders. And so in those areas, we have specific guidelines as to how we can implement control. And it varies by different public lands and whoever's managing them. We have 47 miles of intercoastal waterway. We're surrounded by the St. Johns River as well as the Atlantic Ocean. So we have a lot of freshwater, floodwater habitat and a lot of salt marsh habitat. And our technicians are the bread and butter of our program. They are out every single day looking for standing water. So most of our activities are along the ditches and right of way. Right? So we're treating those on a regular basis, but from an adult standpoint, so I guess I should just give a little basic biology, right? It's only the female that bites. She needs the blood meal for her eggs. The immature stage is aquatic. So our main goal with our program is treating the immature stage. They're concentrated. We have biorational pesticides that we can use in those water bodies. BTI, Bacillus thuringiensis, israeliensis is our main larvicide that we use at our program. And we consider that to be good environmental stewards, right? We want to protect our pollinators. We want to protect public health, human health, animal health. We're not out trying to kill every mosquito that's out there. We're using science to make those day-to-day decisions. This is our science team here at the district. We have a variety of expertise within our program, from engineer, wildlife ecologist, vector biology, molecular biology. And so we have really grown this program. A lot of my staff has started off through grant-funded positions that we've been able to then move into our operational budget and support. So we collaborate with a lot of different funding entities that have allowed us to expand our program and our science program. These are some of our collaborators: the Department of Defense, CDC. We've worked University of Florida, a wide variety of different institutions and universities that we've been able to partner with over the years. And this is just kind of an example since 2020 of some of the grant and then industry sponsored collaborations that we've had. So we work not only with academia, like through NIH collaborations, NSF, but we also work with industry to help we have the capacity at our district to go from intrinsic toxicity in the lab all the way to the field, working with industry to do formulation of products, modes of action. So we really are working with our partners to develop more green products, and we evaluate everything that we use in our program so that we know that it's working on our target organisms as well as, the efficacy of it, trying to reduce, the cost to taxpayers with our program. So I wanted to touch on some of the things that might be of interest to colleagues in the room. And so we do monitor for arboviruses in St. Johns County. And we do this through a variety of ways. We collaborate with the Florida Department of Health and our local St. Johns County Department of Health that are going to provide us information on if we have any type of human cases or veterinary cases, right? So horses, humans, West Nile virus and Eastern equine encephalitis virus are our main arboviruses of concern in St. Johns County because we always have it circulating in our sentinel populations. We're very lucky that we don't have many human cases. In fact, since 2003, 2004, I believe we've maybe had 5 human cases, and 3 of those West Nile and 2 Eastern equine encephalitis cases. So I'd like to attribute that to the excellent mosquito control that we do in St. Johns County, but certainly recognizing that mosquitoes don't know borders, right? So they may be coming in from, from north part of St. Johns into Duval. There's always a lot of transmission of West Nile virus along the Duval, St. Johns County line. But we test, in Florida, we use chickens as a sentinel animal. And so we have 10 flocks throughout the county, 6 birds in each flock that we bleed once a week. And we're capable of doing that testing in-house for West Nile virus. And so we can have same-day results. In addition, we send samples to the Florida Department of Health in Tampa, the Tampa branch, where they also test for Eastern equine encephalitis, St. Louis encephalitis, and Highlands J. And so that information is reported back to us, but we use that information to make control decisions. And so, again, having same-day turnaround, especially for West Nile virus, if it's circulating in our county, then we know where we need to implement control. We also have traps where we monitor mosquito populations. How do we know when to control? Again, I mentioned we have about 50 different types of mosquitoes in the county. We might get a trap with 10,000 of one particular species, but it doesn't feed on humans and it's not maintaining, virus in the enzootic cycle. So we don't need to implement control, right? So we use that surveillance data as well as our arbovirus surveillance data to make decisions on where we're going to implement control within the county. And I really like to highlight that because, again, I think historically people think, even when people call, they're like, can I get on the schedule? Can I get on the schedule to be sprayed? And that's not how we operate. Certainly, we'll take it under advisement. If you think you have mosquitoes, then we can come out, do an inspection, and make a decision on if they are mosquitoes and if we need to treat. But a lot of times, mosquitoes get confused with other insects, other dipterans, other flying insects. And so we do have to verify that they're mosquitoes and not midges and no CMs or INATs. This year, when we were really dry, INATs were a very big issue for us. We got calls almost every single day, but that's not something that is within our mission and that we're able to control for. So part of the surveillance here, you can see we take a blood sample from the wing vein, and this is our laboratory space where we can set up the testing for that. We have protocols for if we have reports of potential local cases of dengue in our county. We can test for dengue, chikungunya, and Zika. We also have a trialplex for West Nile virus, SLE, and Eastern equine encephalitis where we can test our mosquito pools. And this is just an example of a collection that we would have to sort through, determine which species we want to test and which ones we can just kind of toss, if you will. These are our chicken sites currently in St. Johns County. And then this is how we house them. And this has been a big change. One of the things that, this might not be that appealing, but we used to house them in individual like metal cages. And I think from our perspective of our staff and the morale and dealing with chickens, we have really gone above and beyond to try to improve that program, both for the quality of life of the chickens as well as the quality of the life for the staff who have to work with the chickens, right? So they have 1 year service. They get a 1 year public health service that they put in, and then they get to go be free. We place them in good homes, people that want them, that want to take care of them, and they do produce a lot of good eggs. So we're lucky from that perspective. But we have at our site, we have a chicken house where we have to maintain them because we get them, I think they're about 20 to a little bit older, 20 weeks old when we get them. We have to bleed every single one of them to make sure that they haven't been previously exposed. And then they're housed in an indoor chicken building so that we know that they haven't potentially picked up any of the viruses while they've been housed before we put them out in the field. So as mentioned, the Florida Department of Health, they do Eastern equine, St. Louis, West Nile virus, the different assays that are performed. And this is what, when we send those samples to the state lab, these are the tests that they're performing. And that's how we get a confirmation, right? So if we test a bird in-house and it pops up positive for us, we still have to get that confirmation from the state for it to be reported through the through that arbovirus reporting that goes out each week. But for our purposes, even if it is a false positive, it's better to be proactive and preventative in the case that it might be an actual positive. So even though we don't have a state confirmation when we test in-house, we're still going to implement control as if it is a positive when it pops positive. And so we have our in-house and our previous molecular biologist who gave a talk a couple of years ago here. He developed this in-house testing for us during COVID because we're dealing with RNA viruses, right? And so at that time, all of the testing material was being shifted to COVID. And so we still needed to be able to do the testing to protect, our citizens in St. Johns County. So this just kind of breaks down the comparison of our testing. And I'm not going to labor too much on that, but if you guys have questions, I can send you to the appropriate sources. But this is just kind of breaking down the summary of what we've seen so far this year. We've had a total of 14 West Nile virus positives, and of those, 11 have been confirmed. And the confirmations with the state, it's 2-week process, right? So they want an initial bleed, right? And then it's suspected positive, and then that second confirmatory bleed and testing, and then we get that positive result. So we're working with 2 weeks with the state versus same day in our lab, at least for West Nile virus. We've had 2 Eastern equine encephalitis virus seroconversions in our sentinel chickens. And then we did have a positive human case this year for West Nile virus, but it was an asymptomatic blood donor. All right. So, and that was really early season for us. Typically, we start seeing West Nile virus activity in our county more towards like August. And so this, I believe, was like in June. So it was really kind of surprising to us. And really, our chickens, you can see here, didn't really start picking it up in our bird population until September, or end of August, early September. And I just wanted to kind of break it down from a state perspective. Certainly, we see local dengue transmission in a lot more of our counties here in Florida over the last 5 or so years. And so it's really something that we need to pay attention to. In St. Johns County, we are actively working within our urban kind of communities for prevention and control of the vector Aedes aegypti, which is involved in dengue transmission. And then I wanted to touch a little bit. So dengue is an issue in Florida, but also we have Oropouche virus. And so I was asked to kind of touch on this. So last year, we saw a large kind of epidemic coming out of Cuba. And what that translated over to is we were seeing travel-related cases in the state of Florida. We didn't have any locally reported cases in the state of Florida, but certainly we were concerned. Now, we're very fortunate that mosquitoes have been implicated, but there's not been a lot of research into Oropouche virus. I mean, we all know in this room, funding drives a lot of our knowledge, right? And so this really hasn't been on the radar. But there has been work actually done at the CDC maybe about 4 years ago. An entomologist was interested in like better understanding Oropouche, and it just so happened now she's one of the leading experts. She was doing her postdoc as a fellowship with the CDC, another University of Florida grad who did her work down in Vero Beach. But we're lucky in this year that we haven't really seen the same number of travel-related cases. In addition to that, culicoides have been kind of the identified vector. So no CMs, right? We're very fortunate that the coastal culicoides that we have has not been implicated. It's kind of been excluded in some of the vector confidence studies that has been done. So what I like to say for us that are, in Northeast Florida along the coastal areas, we should be good with, not seeing any local kind of transmission of Oropouche virus. Again, more work is needed. This was kind of being spearheaded by the CDC through funding to University of Florida, but we all know where that stands at the current moment. So I'm not really, and in fact, one of the CDC fellows had just basically come up with like testing guidelines and protocols right as soon as, all of our partners were getting laid off, right? So the person that developed the protocols and the testing never got to publish it officially through CDC, and they were laid off simply because they didn't, they were at the end of their, probationary period. So, however, I'm hopeful in the future we will see it, right? So, but we do know ceratopagonids, so they are very tiny, right? Fighting midge, extremely high populations. They can be found inland and along the entire coast of Florida. Typically, so one of the hardest things with our culicoides is the issue is that we just don't have a really good idea of where they're breeding, right? They're not like mosquitoes utilizing water. They're found in soil. Well, good luck, So even implementing control would be challenging. But we do have partners in Lee County, Florida. They have worked and evaluated Sanibel Island, Captiva, high populations of culicoides. They've done a lot of work with aerial applications of the products that we use in mosquito control, and have found that it does a pretty good job of knocking them down. So if we had to develop and implement control measures, we have at least kind of a framework to work from. So as far as our science department and talking about some different novel control methods, one of those is utilizing our drones in mosquito control. And so we have a drone currently that has a 40-pound payload. So it's in total, it's about 70 pounds when it's loaded with pesticide. It's for larviciding. So a lot of times finding or accessing areas that are holding water. In Florida, we like to develop right along the marshes, right? So you're right along that coastal area. There's no real buffer. So getting back there to treat the salt marshes can be very challenging. So we have utilized drones and our aircraft to target those areas. But some areas are just not quite large enough that we would want to send out a helicopter to treat. And then we could utilize the drone to be able to use and target larvicide interventions. I don't know if you guys have seen in the news where in Hawaii, they're targeting malaria, avian malaria, and they're using sterile insect technique, which I'll talk briefly about, but they're releasing the mosquitoes via drones, okay? So it's really cool. There's a lot of potential utilization of drones. We have a LiDAR system where we can look at the difference in elevation and where places might be holding water, right? So we're able, I think I have a couple slides. This is just an example of an area that we might treat. So this is a dip of a salt marsh, larvae wiggling around. Here you can kind of see a boil of mosquitoes, if you can see it real close right there. And then this is another way that So for example, if with a drone, we don't have to be out here walking the field with a backpack, right? So you can imagine that there are safety concerns with our employees being out in the heat, the elements, snakes, gators, you name it, ticks, tick-borne illness, certainly, we want to protect ourselves from. And so the drones have allowed us to be able to kind of target those other areas. Again, utilizing LiDAR, we can kind of pinpoint areas where we anticipate, we have this whole area that gets inundated, right? But where's the water mostly concentrated, and where do we need to target our applications? And so we're looking to utilize this even more. And so one of the studies that we did is we took a couple different areas, ran the LiDAR over those areas, and made decisions on how we were going to treat and evaluate different products within those treatment areas. And we did before and after trapping. We didn't really see a significant impact on our application, right? Like whether we treated the entire area versus using that LiDAR data, the imagery to like pinpoint the areas where we should target. But certainly it's something we want to investigate further. So this was kind of a quick and dirty type trial, preliminary, just getting our feet wet with running the LiDAR, because there's a lot of data processing. And so I don't even tend to tell you that I understand all that's involved in that, but luckily I have really great staff that have some understanding and are willing to kind of dig deep and learn how to take the images and the LiDAR and overlay it and get those maps. So what we did find was that focal treatments of larvicide on targeted breeding sites is not a significant control measure, but we still need to investigate this further. We're also looking at different products just because of the way the drone puts it out. Some provide a longer life of the drone because the products can be a little corrosive on the material that the drone's made out of. So as I mentioned, sterile insect technique. In St. Johns County, we're also implementing sterile insect technique. So we built a 6,000 square foot facility where we can irradiate irradiate mosquitoes, the male mosquitoes. And this is just I think it's a cool photo. We were doing mark release recaptures of our irradiated males. And this was a cool photo that we took. So the way SIT works is insects generally only mate once. And so if a female mates with an irradiated male, a sterile male, she thinks she's mated, and then she goes on to lay eggs that are unviable. And so this is a species-specific approach, okay? You have to have the perfect target organism. The reason Aedes aegypti, our urban container breeder, dengue vector, globally spread, she's a good candidate for this for many reasons. One of those being that she does not fly far, all right? She flies about a mile at most when she's looking either for a blood meal or to lay her eggs. So she's very concentrated in areas. Typically, there's been a lot of studies that they won't cross like major roadways. They stay kind of concentrated locally from where they hatch off from. They also have, they're easy to rear. So if you're going to take an organism and you want to do mass releases of millions of that organism, they got to be easy to rear in a lab, right? You've got to be able to produce them and at a high quality with fitness. And so this species has been colonized forever. We have a local strain that we worked with partners to colonize for us because we're not capable of doing the genetics to make sure that they're super fit. But we have a company that we work with that helps us with that. And then we do all the fitness testing in-house. So, I mean, they got to be as competitive as our native males, right? So they got to get to the ladies. They got to be able to find them before the native males find them. But we have a number of different phases with this program. It's not like we can just go from, 0 to hero. There's a lot of processes involved and quality control measures that we have to ensure that are in place so that we can repeatedly be able to, put this product out. And so one of the first things that we did was we had to determine the diet. We have 8 insectaries at our district, so we rear a number of different mosquito species. We have, of Aedes aegypti, we have 3 different strains. We have our Aedes aegypti strain, we have the Orlando strain, which has been colonized since the 50s, and then we have a resistant strain, because when we talk about pesticides, resistance is one of the biggest issues that we face in the future of control, because we're a small industry, public health, right? If we talk about urban pest control, they have a lot more money in their industry to produce new chemistries, to get new products on the market. For us to get a new product on the market is anywhere from 100 to 150 million, right? And being that we're public health, the money isn't this. I mean, I pay $36 a year for my mosquito control. So if you live in St. Johns County, look at your bill, you'll see mosquito control. I pay $115 a month for pest control, right? So it's the best bang for your buck. But anyway, so we had to go through and select the diet. Then we had to identify the radiating dose, right? So we can't just base it off of other programs. We have an X-ray irradiation machine at our facility. And so we had to work through and figure out what is that irradiating dose where we still have really fit males. They're sterile, right? They're sterile, but they still can compete. In addition, we needed to know, what is the ratio that we need of our sterile males to non-sterile males and females, right? So how many males are we going to need to release based on what's in the natural population? So we had to kind of recreate that. So our ratio where we think we'll get really good control is 5 males to every 1 wild type male, okay? So in order for us to know that, we also have to do a lot of surveillance in the areas that we're targeting. Currently, we have 3 sites that are about 25 acres each, where we're doing these studies, as well as starting to do suppression releases. For us, Lee County Mosquito Control has kind of led the way, and they release close to 1 million male mosquitoes a week. We're working up to doing about 100,000 males per week. And it is based on what you're seeing in your own surveillance, right? Like more probably is better, but you have to take into consideration labor, money for the program. And so for us, we're sitting at about 100,000 males a week is what we're going to be targeting for next year operationally. The way that we understand the mosquito movement and how many we need to be releasing and how many points do we need in each one of those sites, right? In each one of our sites, how many release points of the males do we need in order to get that site saturated with our mosquitoes? And so we do what's called mark release recaptures. We release them at certain points. Again, this comes back to those dyed males. And so we know based on when we're recapturing them, the color, right, that they're dyed, we know exactly where they were released, and we can tell how far they're flying from that release point. And so those are things you're looking for. You're also looking for how long are they out there? How long are they going to be still flying around after you release them your initial time? And good to say that we got 12 days out of our males, which is fantastic because it was really hot when we did our first mark release recapture. And so they're very fit. We're very happy. And then obviously we have to engage the community, right? When we're implementing new strategies and new tools, we can't just go into these communities and not provide information, right? Because what happens? They see you releasing mosquitoes from your vehicle. What do you think is going to happen, right? Bill Gates is releasing all the mosquitoes, he's modified them all. So we have to be working there the entire time that we're doing these releases. And so we've done a lot of canvassing events. We've worked with a social scientist at University of Florida to ensure that we're communicating, the way that we need to be communicating, that we're asking the right questions. And interestingly enough, the communities that we're working in have been really receptive to our program. And they're also, they lean towards like GMO, because that's in the future. Genetically modified mosquitoes will be the future, whether that's 10 years from now, 20 years from now. Oxitec is already working in the US in the Florida Keys. They're global. They have products that you can buy. You know, if you're in Brazil, you can go to their version of Walmart and get a box and put it out and rear your own mosquitoes that have the genetic modification. So we're also trying to understand. We're not going out into the communities and saying it's a bad word. We just want to know where they're at, what the temperature is. It won't be us leading the science, right? We're the end user. We can evaluate it for them, but we're not the ones driving that science. But we want to know what you know, how to prepare for the future, right? So we're asking some of those questions as we're talking about sterile insect technique. One of the cool projects that we have with University of Central Florida is working with a professor there that has this. It's called cap gel, and it's a collagen alginate based self-assembling biomaterial. And it was originally developed as a capillary hydrogel scaffold for tissue regeneration. Well, so we've collaborated with this scientist who has made this material for us. So one of the things that we would love to do is get rid of our Sentinel chicken program. All right. We love the chickens. We want them to be happy and healthy. It can be quite draining, you know, leaving them out in the field. But luckily now that we've changed some things and they're at the fire department, a lot of our Sentinel chicken sites, and they love the chickens there. Like they'll have them out while they're working out. Like they're living a good life now. But we would like to get rid of this program. And so we've partnered with UCF. We have a grant through the Florida Department of Agriculture and Consumer Services. So what we're doing is we're filling these cap gels with blood, and we're seeing if we can pick up mosquito feeding as well as virus from these cap gels, right? So we've done some infection studies with University of Florida where we've taken, I believe, Senbus virus, introduced it, and then we're able to detect it in the mosquitoes, right? So what we're thinking we could do is put this material in our traps that we put out every week, right? And they're going to, when they feed on it, they're going to release the virus into that material. And we're also doing microsatellite DNA testing to determine if we can identify the species of mosquito that's feeding on it. So this is some of the work that's ongoing in our lab. We've had to do a lot of optimization because right when we got this project, our molecular biologist left. And so I've had two really great technicians that have been able to kind of take this project and run. But it has a lab component and then a field component. So we've kind of completed the protocols that we need, right? We've tested those, we've optimized those. So now we're taking it into a semi-field setting in these cages and putting different traps out in these cages and releasing our mosquitoes in there, and then seeing if they're going to feed on that cap gel that's in the trap, right? And so there's a color, you know, you put a dye in the cap gel, and you can visually see it in the abdomen of the mosquito. So that way we know that they fed on. Right now we're working with blood, putting bovine blood in the cap gel, but there's another project that's just taking sugar, right? Mosquitoes, only females take the blood meal, but they need sugar to fly, to mate. And so they do a lot of sugar feeding. So if we put sugar in those cap gels, can we also be able to detect virus? Because that'd be a lot easier than, you know, having to go through a blood, you know, supplier to provide you the blood each week to get it out. And also it doesn't dry out as much with the sugar water as it does with the, so. And then, so I think I've hit all three of the questions that I submitted for you guys with your QR code for your CEUs. But the one thing, you know, that we can all do, and the best information that we can give, you know, to our neighbors, our friends on how we can prevent disease transmission is by utilizing repellents, mosquito repellent. And so we have this cute QR code here if you want to find your match, because I will never recommend anything to you that is not EPA approved, right? So because I'm talking about disease prevention, I'm not just talking about the nuisance of mosquito bites. And so we're going to recommend EPA approved, CDC recommended repellents when we're talking about disease prevention. And so you can go here to find, but DEET's the gold standard. Picaridin is really good as well. And they have new formulations where it goes on dry. The one thing that I didn't put up out here, this was created by, so we have a disease vector education center at our district. It is a 6,000 square foot museum, a very interactive, engaging material that talks about mosquitoes and prevention, but also has a slew of information on other insects like pollinators. We have an observatory beehive inside. We have a fish tank that you can feed mosquito larvae to, and we talk about, predator behavior. We have hissing cockroaches. It is very engaging. It's free to the public. It's open Wednesdays through Sunday, 10 to 5. And we have a great educational staff there. They created this graphic. So if you're ever close to 95 and 16, if you ever go to the outlets, we're right across the street, okay? In our education center. And if you want to come on a Monday and Tuesday, I know somebody that can walk you through. So I highly encourage you guys to come visit. If you have any questions, I'll take those now. Sure. Okay, I love it. Yeah, so not historically. Currently, one of the projects that we have that we're working with Florida Fish and Wildlife. So I mentioned the conservation areas. So we have Anastasia State Park. I don't know if any of you guys have been there, but it has Conch Island, and it's one of the, when that hatches off, every single person in Davis Shores calls in. The mosquitoes are very challenging to control once they hatch off, but it's about 700 acres that we can't access. We have to take four-wheelers down the beach, which is a lovely day, honestly. But we can only treat about 10 acres of that 700 acres by foot. And it's really restrictions through the park system. But we could use a drone, right? We have vision of sight, line of sight. We could utilize our drone in that area. But one of the biggest issues is the bird nesting season. So our shorebirds, right? And so that just happens to coincide May to September, which is exactly when like the height of our mosquito season is. So we're working with them because they've asked us to take some classes on the shorebirds, which we will gladly do, love to learn. But one of the things that I posed to them was like, well, how, when these mosquitoes hatch off in the millions, how is that impacting the shorebirds, right? Like it has to have some sort of fitness impact because these mosquito species, to your point, are, they're game for anything. You provide them a buffet, they're not picky, okay? So I would assume that they're feeding on some of these shorebirds that they're trying to protect. So that is a project that we've lined up for next year, is we're going to do some testing of the mosquitoes from that area to see, what are they feeding on, right? And we know the shorebirds in that area, right, of interest. So we can use those primers, do blood meal analysis on the mosquito bird population. Well, so bats in general are opportunistic, right? If they're insectivores, they're opportunistic. They're not just honing in on mosquitoes. So actually from the scientific literature, it shows that mosquitoes are actually just a small portion of their diet. But we have worked throughout St. Johns County putting out bat houses, right? Unfortunately, we put out 15 of them in 2010, and we only had one where bats actually established in those bat houses, right? But at our education center, we do have a bat house, no bats, but we have a lot of information on, opportunistic feeding behavior of some of the predators, because a lot of times we'll get people calling in and saying, oh, bats and dragonflies, that's all you need. Stop spraying pesticides, right? Well, we want to work with nature. We want to work with our predators, but it's not a one size fits all. We still have to incorporate other methods of control. But from our perspective with bats, we've certainly had issues of trying to get bat populations established in the bat houses that we have put out through St. Johns County. And for us, we don't see it as a good control measure, at least in our area. So did you have anything you wanted to follow up with on that?
No, I haven't.
Okay, okay.
Is that a predictive thing that you have a window of opportunity to sort of shut things down? How does that compare to when we hear about the wildlife birds that they say a lot of dead birds don't touch?
Right.
Like, is there that sort of wave?
So yeah, so if we remember in 1999 when West Nile virus was introduced into the US, right? One of the indicators was crows were just falling from the sky, right? But now they don't, we don't really see, they've built up, they no longer have that susceptibility really, right? So our wild birds aren't a great indicator unless we're misnetting them and bleeding them and testing them. So that's where our sentinel chickens come in. And it does provide us, an opportunity to get out in that area, identify the breeding sites that are, so Culex, our West Nile virus vector, she doesn't fly very far either. So a mile, 2 miles. So what we can do once we see the circulation is we can even go in, look at the maps, right? And determine what bodies of water are breeding. And it's very specific, like stagnant water. So ditches, wastewater plants, right? We can go in and make those treatments. So yes, it does allow some sort of like preventative measure. And we certainly want to try to prevent it before we see human cases, right? So that's kind of the last type of sentinel. I mean, we're sentinels too, right? But that's not the sentinel we want to be using. So, yeah. So we're only in St. Johns County because that's, our tax base that pays for our program. But we do collaborate with Duval. We do collaborate with Lucia, East Flagler programs. For example, Duval has a really great entomologist, but their program has been like cut and cut and cut. But he's very interested in dog heartworm. And so he's worked really closely with like the animal control in Duval. And so we've actually provided and tested mosquitoes for them, right? So while we don't, we're not doing surveillance for them, if they have mosquitoes, we can test them in-house because we can test in-house also for dog heartworm. It's a DNA virus versus RNA. But we had a project in St. Johns County as well, interested in identifying what species are involved in dog heartworm transmission. So we do try to collaborate. So we have memorandums of understanding. And if they were in an emergency situation, then we certainly would, do our part to help out. So, yes. Oh. Right. So yes, it is a huge labor and cost investment for the sterile insect technique program. A lot of what we have conceptualized with this program is through collaboration. For example, we have Mosquito Mate, which is a company in Kentucky. They have a registered Wolbachia product. So if you're familiar with, they have it's registered as a pesticide. It's a biopesticide. But they use Wolbachia, which also induces sterility when the Wolbachia males mate with wild type females that don't have the Wolbachia. So they're utilizing some of our lab space, our insectary space. And so with that, through that collaboration, that brings in personnel that we can leverage, right? So they may provide their salary, we can provide their benefits, and then we have, maybe 30% of their time can be contributed to our program. So that way I don't have to, hire a full-time individual, although I'd like to get there, but my boss and I have a little bit different idea of how we're going to get there. But yeah, it is going to take personnel. It is going to be a huge investment. But certainly, since we start to see dengue increasing local transmission in Florida, chikungunya this year, 2014 was a big year for chikungunya. We could potentially see another outbreak of chikungunya. I think that we're driving that science and we're driving that technology, but that space was more developed for collaboration as well. So we have WHO, who has identified our site as a training site for like Latin America. Originally, I think we had hoped that we would be able to like produce them and ship them, but the logistics of that, like producing, like Google has done a huge investment. So Miami-Dade, they have like a 30,000 square foot building where they're doing this. And they're shipping to Latin America. It's just not something, under our capability. But I do think that it has utility, right? Aedes aegypti is highly resistant to pesticides, to a lot of the different pesticides that we put out. And this was one of the issues in Miami-Dade during Zika. They weren't, the CDC was there. We had industry there. We had emergency responder programs that work in mosquito control there. And they weren't able to knock down Aedes aegypti populations because they were so resistant to mosquitoes. So they developed a new larviciding strategy. So we had a new tool come out of that, but we don't want to be in that same situation in the future, right? We need to have different tools available for us to incorporate in our programs. So yeah, it is a huge money investment compared to traditional, traditional pesticide. Our budget for all of the larvicides and adulticides that we use is probably close to $1 million just for those pesticides. I would say for our SIT program alone, we've invested more than that, and we'll have to continue to invest in it because the technology is only going to change. It's going to become more advanced. So, sorry.
So I'm going to take place this question. The first one has to do with irradiated males. You leave them in the area with the host of and so, question was brought up. Everything has sort of a niche in ecology. What would happen if you were somehow able to eliminate all?
Never happen. It would never even be able to happen. So as I mentioned, we have over 3,000 different types of mosquitoes that utilize a variety of different habitats. Some mosquitoes develop in crab holes, all right? Some mosquitoes only feed on annelid worms. So mosquitoes do provide a lot of food for opportunistic insectivores, so different birds, bats, fish, the immature stage. Other, organisms may feed on them in the water body. So they serve some role, right? But when we talk about eradication, we have to think of it on a species by species level. And as I mentioned, Aedes aegypti, it's not like we're going to be blanketing all of St. Johns County. Aedes aegypti isn't even distributed throughout the entirety of St. Johns County. It's very coastally focused, right? So the further inland you go in St. Johns County, they're just basically not there, okay? So, when we talk about these targeting mosquitoes and control, it's even our truck-based methods, you get 300-foot swath, whichever way the wind blows, right? You have vegetation, you have houses, you have fences, everything like impacts that penetration into the area that you're targeting. So, and you've got to get them when they're flying, right? Our adulticides only work if the mosquito's flying when the pesticide's being applied, because it's not supposed to provide a residual. It's not an agricultural application. We're not applying it to a crop hoping that the insect feeds on it, right, and kills it. It's meant to not provide a residual because we don't want it getting in our water bodies. So, you know, it's really a shot in the dark that we get such good control whenever we do it. But the science has, you know, kind of changed how we apply, how we deliver it, the formulations, the droplet size. But yeah, I don't think you would see like this huge crash in the ecosystem, because I don't think, I mean, it's even hard for me to go theoretically there because it's just not possible at all. I wish, you know, we could like knock out a certain species and not have to worry about, and maybe someday we will be there with like gene drive for Aedes aegypti. But I think that there would be ethical considerations to try to do something that would be like just blanketed eradication of every single species, right? So because they do play a role. So, sure. Oh, well, so it's yeah, so it is Oxytec, they're friendly mosquito. So essentially they have engineered it so in the presence of a certain chemical in the larval stage of its rearing, it turns a gene on, right? That basically makes it, when it mates with a female, uh, she doesn't have viable offspring. Okay. And so, and they've modified this over the last like 15 years. So this was started in 2010, just trying to evaluate it in the Florida Keys. And in 2020, they finally got the buy-in, right? But this product has been distributed throughout globally, Asian market, Latin American market. Brazil has been like kind of like leading the way with their case studies. But it's essentially you go to the store there, you buy a box, you add the components and water, the larvae develop, and then the male mosquitoes fly out. They go find a female, and then you don't have viable offspring, right? Um, there is a company in the US that's also working on, they're using a gene that cuts off flying in the females. So the males, so it's a box idea again, you add the water, but when the females hatch, they can't fly away. So they still like get out, but the male is sterile, that gene, you know. And so they fly away, mate with the wild type females, and then you have suppression, right? So that's something that's getting like we'll be doing experimental use trials with EPA and this company coming into, well, hopefully this summer. We'll see where it goes if it gets moved forward. But that's called NGSIT. So with that company, the Oxytec, the trials in the Florida Keys went really well. But one of the biggest issues with a lot of these trials is if you have a hurricane coming in and you have protocols. And so during the years that they were evaluating it, we had a number of hurricanes. And so every time it'd be like they get it put out, ready to go, 2-3 weeks going, storm comes in and you gotta, yeah. So, but I would say within the next couple of years, you'll see something on the market that will be for homeowner to distribute in their yard. And then we'll certainly work at the public health level to potentially incorporate it, but it may even go private pest control, because private pest control has gotten into the space of mosquito control as well. So, but I would say very soon we will have the capability of purchasing our own, grow them at home, and hope they find the ladies. So.
Any more questions?
Well, thank you guys for having me.
Dr. Pauls, for a great presentation. Mosquito talk is always so fascinating to me. Thank you to everyone for joining us tonight in person and virtually. And once again, thank you, Tim and Byrad, for supporting JAM. So our next meeting will be Tuesday, December 2nd at Baptist. And the presentation title is STI Testing Recommendations. This will be presented by Dr. Mondrea Howard. And for more information on the rest of the schedule, visit us online. And once again, if you need to pay for JAM, email Megan, or you can give me your checks or pay online. All right, you guys have a good night.